So, Dr. Tom Madden, we're going to be interviewing you today, the founder and CEO of Acuitas Therapeutics. You and your team, you guys have made amazing strides in lipid nanoparticle technology. This has allowed for a lot of mRNA therapeutics to be effective, including vaccine delivery with the Pfizer-BioNTech COVID-19 vaccine. Amazing work you guys did helping us to pull us out of that public health crisis. It's an honor to be here with you today. You're really pioneering leader in this field, and looking forward to getting started. So I wanted to just go over quickly, if you could give us like an overview of your career background. I know you had a journey from academia into biotech, and I was wondering if you could tell us a little bit about how that went and maybe the challenges you faced along the way.
Sure. I started off, very much on an academic path, undergraduate and graduate degrees in London, England, and then had always wanted to come to North America. Applied to come to a couple of different places, mostly in the States, but I got a fellowship to come to UBC, and I came out here in 1980, having no idea what Vancouver was like. Could have been anywhere. Could have been Cleveland or Detroit or anywhere. And so I was, shocked at how beautiful Vancouver was and the campus. And so although I originally imagined I'd be postdocing for a couple of years and then go back to England, that plan quickly changed, and I decided I'd be happy to stay here. And the postdoc work I was doing was far outside any immediate, clinical application. We were doing biophysical research on cell membranes, but we needed to develop model membranes in order to do this work. We came up with a new technique to generate model membranes of a very defined size, a very convenient size. We then realized, almost serendipitously, that those liposomes could be used to deliver drugs. We realized that we could create conditions where you could very effectively load drugs inside these liposomes, and then change the drug pharmacokinetics and biodistribution. And we felt that this had potential clinical applications. And we were just fortunate that that work was ongoing in the early mid-80s, at a time when the whole idea of starting biotech companies was really just getting started. At that time, nobody, or virtually nobody on campus, would be collaborating with a pharma company or a biotech company. But we, or Peter Cullis, who was the head of the group, was contacted a biotech company in Princeton, New Jersey, who were also developing liposomes for clinical application. And we quickly created a collaboration where we could continue our academic research, but at the same time be able to contribute to the development of the drug products that this company wanted to take into the clinic. So it was a sort of an ideal collaboration that would support more work in the lab and allow us to continue to do what we were already interested in pursuing.
Wow, that's pretty amazing. And I guess kind of going along with that theme of translating academic research into viable technologies, I think that's something a lot of students are interested in, and something that's happening a lot more frequently nowadays. And I was wondering, what advice would you want to give to students who are interested in going down that similar path?
I think it's. I think the interface between academic research, basic research, and a variety of different applications. Whether they be clinical, commercial, environmental. As you say, is really growing. I think. For a student in a lab, their program obviously is largely determined by their supervisor, and so, if a student has an interest in applying their research or potentially applying it, then I think obviously they need to look for an opportunity for that to happen, and maybe pick a group that is interested in sort of external collaborations like that, and I think also sort of be open-minded in terms of what those potential applications could be, because certainly in our case, as I say, we were doing very basic research, biophysical research, and it was serendipitous that we recognized that there was a potential, clinical utility for that work. But it takes sort of like an open mind, to be able to appreciate that there is that possibility.
For sure, I guess kind of you're hitting that point. You have to always keep your mind open where the research could take you, and also be part of a lab that might be collaborative in that way that would allow you to take advantage of those opportunities when they come your way. And one question I had, I'm not too familiar with the whole process of how this works. I was wondering, so if you have an idea that you think you might be able to translate into a product that you could scale on the market, how does that work going about? Do you have to purchase the intellectual property from the university, or how does that work?
That's a complex area.
Okay.
Generally, if you're working at a university, if you're funded by academic grants or fellowships or whatever, then the work you do there, the intellectual property you generate, is owned by the university, or at least in many instances that's the case. Obviously, you would be potentially an inventor on anything novel that you discover, but I think that the ownership wouldn't necessarily rest on the student inventor. Universities typically will have, as UBC does, a university industry liaison office, and they certainly encourage researchers who think they have something patentable, to go to them, and usually the university will facilitate the filing of a patent application if they agree that there's something that should be protected. What happens to that IP afterwards then is potentially a bit of a grey area. The university could have an outreach program to companies, making them aware of the IP that they hold. Probably more often, or at least as often, the individual researchers would then look for opportunities to create a connection with a biotech company or a pharma company, and see if they would be interested in using that IP. But that would then require the university providing a license to that company in order for them to be able to take advantage of that novel invention.
I see. I think we're really lucky to be able to have those opportunities here, because myself, a lot of my students, we are involved in research here at UBC, and you never know what could happen with the research that you're doing. So I'm really glad to hear that we have those opportunities, that in case something novel we came up with, that we could actually scale it and get in touch with these companies and turn it into something bigger. It's really inspiring, honestly. And then going along with these novel technologies, I wanted you to kind of give our listeners a little bit more information about the lipid nanoparticle technologies and why they're so essential for delivering nucleic acid-based therapies.
Messenger RNA, the idea of using messenger RNA for clinical applications really arose in the early 2000s. But there were huge problems that needed to be addressed in order to allow that to happen. One of the biggest challenges initially was that mRNA administered to animals, for example, would trigger a very strong immune reaction. Our bodies, our cells, are designed to identify foreign RNA, typically viral RNA, and then they will alert the immune system, to the presence of a foreign organism generally. And so, if you administer mRNA, it is recognized, or it can be recognized by specific receptors in our cells, as I say, that then initiate this immune reaction, and it can be quite severe. And so it was critical, the research that Drew Weissman and Katie Carrico undertook to identify modified uridine bases that would allow a foreign mRNA to be administered without triggering this immune response. And so their seminal work, obviously recognized by the Nobel Prize, really opened the door for people to now look at messenger RNA as a true therapeutic. But the huge challenge remained was that messenger RNA is incredibly labile. It breaks down very, very quickly. We have RNAs on our body. We have RNAs inside our bodies. And if you administered free mRNA, it would be destroyed within seconds. It's also a huge polar molecule, so it can't get into our cells, and it needs to be in the cytoplasm, in order to be translated. And so you need a delivery technology that will protect the mRNA when it's administered, and that will carry it into the cell and specifically allow it to go to the cytoplasm in order for the potential to be realized. And that's really where LNP delivery systems come in, because you can encapsulate the mRNA inside these microscopic lipid spheres that will protect it. And at Acuitas, we've been able to identify mechanisms whereby we can very effectively trigger recognition and uptake of these LNP into cells, taking advantage of a receptor-mediated process. So essentially we design our LNP to look like lipoproteins, which are taken up into cells in order to provide, essentially, fats, nutrients to the cell. And so we take advantage of that uptake process in order to get initially into an organelle called the endosome. And then again, our LNP are designed so that within the endosome, as that matures, the interior becomes acidic, and then the ionizable lipid becomes charged, and it can interact with the endosomal membrane and allow the payload, the mRNA, to then escape the endosome and get into the cytoplasm. So it's a very sophisticated technology to allow that delivery specifically to the cell cytoplasm.
Wow, yeah. I didn't realize how many steps it would take. Every step you're saying poses a new challenge. First, we gotta get it into the body, then we gotta get it to be recognized by the cell, then uptook by the cell, and then break down properly, all while keeping the mRNA stable, which I know is pretty hard because even in the work I've been doing over in life science, I've been doing some multiplex fluorescent in situ hybridization. That's with the confocal microscope imaging the mRNAs, and I know how tricky it can be to keep those intact, and they're frozen even, right? So when they're in a live body, it's pretty amazing the technology that you guys developed to be able to bring those mRNAs into the cell and to complete the functions that you're desiring. So congratulations. I know it must have been years and years of work that went into that, so it's pretty amazing. And kind of with this LNP technology, I know you guys were heavily involved with the COVID-19 pandemic, and thank you again for the work. Really amazing work that you guys did helping us to bring us all out of this pandemic. And I was wondering if you could kind of go into how that looked for you guys, how you guys got involved, because I imagine it was probably a pretty crazy time, and just give us an idea of all that.
Sure. So we've been working with various partners around the world, prior to 2019, providing them with our delivery technology for a variety of different types of therapeutics. And that also included work on mRNA vaccines. And one of our partners at that time was using our delivery technology for a rabies vaccine that had gone into the clinic in a phase 1 study in 2019. And they released the results of that phase 1 study in January 2020, showing that the rabies vaccine worked remarkably well at an incredibly low dose. And so we were already thinking about to what extent could messenger RNA vaccines be used to fight the pandemic. And so this clinical data was then released just as the global threat from COVID-19 was becoming apparent. So we quickly reached out to CureVac, who were a German company. And we were also working with BioNTech, another German company, again on mRNA vaccines. And so we suggested to both of them that we collaborate on developing a vaccine against this emerging pandemic. They both obviously were supportive of doing that. But BioNTech in particular completely focused all of the work in the company on accelerating development of this COVID-19 vaccine, and basically shutting down all of the other programs they had ongoing, and so that they could focus exclusively on this. So as I say, we worked with both CureVac and BioNTech, but as I say, BioNTech really took the initiative, and so much of the work was with them. At that time, we obviously needed to be able to evaluate different mRNA constructs that they wanted to use to express the COVID-19 spike protein. We also wanted to evaluate a smaller number of different LNP compositions, which we thought would be appropriate for the vaccine. And so we had a very, very intensive program ongoing in the first few months of the pandemic in particular, where BioNTech were shipping out mRNA constructs to us, and then we had to formulate them into our LNP as quickly as we could, and then ship them back to BioNTech for testing. And so this was an incredibly tight turnaround in order to minimize any delays in terms of the development. But we were trying to do this at a time when, first of all, there was a lockdown, so we only wanted essential staff to have to come in to do the R&D work in the labs, and we also wanted to minimize the risk to those staff on duty. So we would have a shift system, so that people would work at different hours during the day and night, to minimize the number of individuals in the lab at any one time. But people were working like round the clock doing this work. The other logistic challenge we had is that the airlines were no longer flying. Nobody was traveling around the globe, and so normally we would just courier these formulations over to Germany for the preclinical studies, but there were no planes flying, so we had to charter flights in order to be able to ship these critical materials to BioNTech for this work. So that was sort of kind of one of these sort of ironic consequences of the global shutdown.
Wow, yeah, no kidding. Working into the two different countries, having to send these delicate materials over across, especially when the airlines are down too. That's a pretty huge challenge that you guys had to overcome. And I think the pandemic in general just really underscored how rapid some of these technologies need to be developed. I would ask you, what kind of takeaways for the future would you and your company have from this pandemic in order to kind of be ready for another public health emergency such as a pandemic?
Sure. I mean, one of the biggest challenges that we faced at that time was the supply of the components you needed for the vaccine, and in particular the proprietary lipids that we developed for the LNP composition, because although for a vaccine, all of the critical components, all of the components in it need to be manufactured under very stringent quality conditions. We were fortunate in that the composition of the vaccine we were using, we'd already commissioned a contract manufacturing organization in the States to produce those proprietary lipids under CGMP, but they were making tiny quantities, and we had the good sense, as soon as we recognized the opportunity to work with BioNTech and CureVac on the vaccine, we immediately reached out to the company that was making the lipids. We had a personal relationship with the owner and the CSO, and so we got them on the line, and we told them what was happening, and that we would be frankly the world would be needing these lipids in kilogram or ton quantities as quickly as possible, and they immediately agreed to scale up their production, which was a massive endeavor on their part. And as I say, that was just the first part of the global scale up that was needed to supply all of the ingredients and components that were required in order to generate billions of doses of the vaccine that were needed. And so people sort of forget how limited vaccine supplies were at the beginning of even after the approval of the Comirnaty vaccine, supplies were really limited. But within a very short period of time, those global supplies ramped up to address the need. And again, that was a phenomenal effort over many companies in many different countries, sort of coming together in addressing that need. And so based on that, we were obviously in contact with the Canadian government. They were interested in the work we were doing to support the development of the vaccines. They also asked us for our advice in terms of preparing for a future pandemic. And so, part of the advice we provided is stockpile the components you're going to need in order to be able to manufacture a new vaccine in the future. Because the versatility of mRNA technology allows you to create an mRNA construct to express a protein on a newly discovered virus very quickly. You can do that in a matter of days or weeks. And so the technology allows you to be able to quickly come up with a vaccine candidate that's uniquely tailored to any emerging virus. But what you need to then ensure is that you then have all of the other components you need to be able to manufacture that vaccine to address the quantities you need, for example, to protect the Canadian population. So our advice is just ensure you have a stockpile of components, but you also have the technology available at manufacturing facilities within Canada to be able to generate the vaccine as needed.
Definitely, I think that's great advice, and I hope the government is listening to you guys and having those ready, because we never know when another emergency will come about like that, because we have such a huge population on this earth, and you never know, one virus might mutate the wrong way and we have billions of people getting these diseases. But I think that's a strength of your guys' company, as you said, with this new mRNA technology. It's pretty amazing that you have the technology with these lipid nanoparticles that all we need to do is sequence the DNA of these, the virus, and then we can develop rapidly these new vaccines and be able to treat people much faster. And with this technology, I was wondering if you could kind of give us an update on what kind of diseases or what areas are you guys looking at now after the COVID-19 pandemic?
Sure. I mean, obviously the success of the COVID-19 vaccines triggered a lot of interest in using mRNA for other vaccine applications. And so, working with our partners, they already have flu vaccines in the clinic with our technology, combination flu, COVID vaccines, RSV vaccines, and a number of other vaccines against fairly conventional viral infections. But one of the beauty of the technology is that you can use it to express multiple different antigens from a single virus, or multiple variants of an antigen from different strains of a virus, and so this opens up the possibility of being able to generate vaccines which are much more effective against in preventing disease. And along those lines, we're working with our partners on both a HIV vaccine and a malaria vaccine, as I say, which really takes advantage of the ability to exquisitely tailor the express protein to the particular viral antigen that you want to protect against. And what's sort of interesting, if you look at both malaria and HIV, around the world you've got different strains of virus or different strains of malaria, which are prevalent in different geographical areas. And so, again, the versatility of the mRNA technology would allow you to tailor a particular HIV vaccine, for example, to the strains that are prevalent in Southeast Asia or Europe or somewhere else. And that way, you're not having to compromise in terms of having to come up with a global vaccine which does a so-so job in protecting populations against those different strains, but you're able to develop a vaccine which is as effective as it can be in each of those different geographical regions.
That's pretty incredible. And so would you say that's due to the availability of the researchers to develop these mRNAs to tailor to these different disease types in all these different areas?
It's that. But the other thing is, if you're now delivering multiple mRNAs to express multiple antigens, the concern would be that, well, is the level of expression of each of those sufficient to generate a robust immune response? And so what was needed was an LNP was an LNP that provided higher levels of expression for a given dose. And so over the last couple of years, we have been working on identifying LNP compositions which are more effective than the 315 LNP used in Comirnaty. And we've been able to identify LNP that are 10 to 15-fold more effective. So for the same dose of mRNA, you can now express multiple different viral proteins without any compromise in terms of the level of expression of each of those proteins. So again, maximizing protection while minimizing any side effects from the dose itself.
I see. It was very exciting because I was even looking at some of the work you guys were doing, and some of this stuff almost sounds like science fiction, like gene silencing or gene expression and these different areas. I think that this technology has so much potential for so many different diseases. It could really be a game changer. It already has been, but even continuing into the future. And I was wondering, especially for our readers, which are neuroscience students, where do you think that this technology could apply in neuroscience?
Before I answer that question specifically, I should say that at the moment, about half of the work we're doing supporting partners is developing new vaccines, but the other half, as you were saying, is around supporting gene editing therapeutics, where our partners are using a variety of different methodologies to either silence genes which are involved in disease, or correcting gene mutations where the required protein isn't generated, or using epigenetic approaches to switch on or switch off genes, but without making any change to the underlying genetic code. And so, that's an area with a huge sort of potential to address a variety of different diseases, where again you need to deliver a messenger RNA, which is the gene editing protein, and in many cases you also need to deliver a guide RNA, which is going to then take that protein to the particular place on the genome where you need to edit. So a lot of the work we're doing today is really optimizing our delivery technology for those gene editing applications. And one of the challenges that we face is how to use this technology to address diseases that affect different cells or organs in the body. If you administer an LNP intravenously, then as a particulate system, it's going to stay within the blood vessels until it's able to exit the capillaries, and most of the tissues and organs in our bodies will have a continuous lining to the capillaries, and so it's only in the liver and the spleen and some other organs where the blood vessels have fenestrations, they have gaps that allow the LNP to escape and to access the interstitial space and be able to deliver to cells in that space. So one of the things our partners are always looking for is, can we use our technology to deliver to the kidneys or the heart or someplace else? And sadly, the short answer is no. There isn't a mechanism whereby you can exit the blood supply in those tissues and organs. And of course, the brain has long been the most challenging organ to deliver any drug to, let alone a particulate system such as an LNP. How do you get across the blood-brain barrier? We have done research where if you directly inject into the brain, then you can see expression of the protein that's encoded by the mRNA you're delivering, and so we'd love to be able to access the brain in a way that provides uniform delivery. So if any of the researchers or the students can come up with an approach that allows that, then we'd love to hear about it and love to work with them. But that barrier, remains a huge one in terms of being able to, directly deliver to the brain. Now, whether you could deliver intrathecally and access neurons in the spine, and for example, is something we'd like to explore. It's not an area we understand. Our expertise is not around brain anatomy and CNS anatomy, but certainly we'd love to open up the opportunity to use these types of delivery systems to access cells in the nervous system.
Very interesting. And one thing, I'm just totally throwing this out there. I'm no expert, even close in any means, in this area, but even possibly for the nervous system, maybe even cerebrospinal fluid, possibly a way to get in there, like you said, within the injections. But that's all just speculation. I have no idea.
People have tried that approach. If you can inject intrathecally into the cerebrospinal fluid.
Yeah.
The problem is that the flux within the brain is extremely inefficient, and it's very difficult to get any to get any reasonable level of delivery,
right?
Even through that route. But as I say, if anybody comes up with a technology or a methodology that allows that to happen, we'd love to hear about it.
Have you been in touch with Pfizer-BioNTech Therapeutics here at UBC? I know that I think Dr. Cullis is involved with that one as well, and I know that they're attempting exactly like what you're saying to develop ways that the LNPs could access the brain. But the way you're explaining it makes a lot of sense. As you said, even for typical therapeutics, it's hard enough to get into the brain. So with this new technology, I'm sure that in the coming decades, we could look back on this conversation and say, "Oh wow, wasn't it obvious?" And we'll be treating all types of brain diseases. But I think, as you said, it's important for our listeners, a lot of the upcoming scientists, to kind of think about these things, these new areas where this amazing technology could be applied. Because I mean, it could be an amazing opportunity if there was a way to target these other tissues as well. But that's for a future date. I know I want to be respectful with your time here, so we'll be wrapping up. I wanted to give you or ask you a question. If you have any advice for young scientists who want to make a significant impact in medicine, but they might feel overwhelmed by the challenges of the biotech field, and just some advice on how they can start to get involved.
It's a good question. I do know that there is a biotech program here at UBC, for example, where students will spend part of their time, working with a biotech company, and then part of their time doing, conventional coursework. And we have a lot of, we have a consistent flow of biotech students, come through Acuitas, work with us for 6 months or 9 months, and as part of their overall program, they get placed in a number of different companies around Vancouver. So that obviously provides good exposure to what it's like to work for a company. You also learn about those individual companies. Are you interested in the work they're doing? Is it something you find exciting? If you're not in that co-op program, I don't know to what extent there's an opportunity to do internships, but that would be another possibility. And I doubt there's I think people would have to reach out to companies individually if they were interested in doing that. I don't know if there's any kind of a program that supports that. But I think, as I say, if there is an opportunity, as I say, to go spend some time with a biotech company, that's the best learning opportunity that you would have.
Definitely. And even for students who aren't necessarily in the biotech program, just in life sciences in general, we have the co-op program too, which allows students to get those experiences in various biotech companies. Are you guys involved with the co-op program?
We are. Yeah.
Okay, fantastic. Yeah. And so that's a great way for students to get involved and learn about what's happening and get started on that side of their career. And you, as a leader in this field and a scientist entrepreneur, for students who would want to follow in your footsteps and kind of delve into that entrepreneurial side, what would you say are the key attributes that they should be working on in order to find success in that field?
It's an interesting question. I mean, certainly you want to have a passion,
of course,
to be an entrepreneur. It takes a huge commitment, to sort of take that step. I think you obviously, want to be as well informed as possible about, what's involved there, and I to the extent you can learn more about entrepreneurship, and the biotech industry or other high-tech industries, it's only gonna help. The other thing to sort of recognize is that nobody can succeed by themselves. You're going to need to bring a team together in order to sort of create a company that has the potential to be successful. So you sort of need to think about what are the skill sets you don't have that others could bring to the table, and think about your own leadership skills, motivational skills, management skills, all of the different attributes that you would need to be a successful entrepreneur, and as I say, to be able to assemble and motivate a team to be successful.
And that's one topic I wanted to touch on, is I think at Acuitas you have really put together an amazing team, and the whole culture of your company seems very teamwork oriented, and that collaboration aspect. And I was just wondering if you could kind of tell us how you built that atmosphere.
So when Acuitas started, I mean, the genesis of Acuitas was sort of interesting in that, as you were saying, we had, or many of the founders of Acuitas had worked together in the LNP space for decades, and several of us had been working at another company called Techmira Pharmaceuticals, a company we'd originally founded, back in the 90s, and we were fired from Techmira in 2008, and we were really interested in the research we were doing. We were excited by the research we were doing and wanted to continue it, and so we started Acuitas, then called Alcana Technologies, to continue that research, but also to sort of keep the team together. There were six of us that started started work at the company, and we remained as 6 people for 3 or 4 years. I mean, the company only grew very, very slowly, but we came together because we wanted to work together. We respected each other. We enjoyed working together as a team, and that team, I think, as I say, had the different skill sets needed to be successful. And so, as we slowly grew, we wanted to maintain the relationship, maintain the culture that we enjoyed. And so we were very sort of proactive in trying to put in place systems that really emphasized, respect and collegiality and transparency and so on. And as the company grew, people really sort of enjoyed that culture. And again, it's been something that has evolved organically, but where people have always felt very protective of ensuring that even as we get bigger, we don't lose sight of the culture that we had always wanted to maintain and work in at Acuitas.
Wow, I think that culture that you built is definitely one of the main drivers of your guys' success as well. I think when you have a team that wants to work, they want to show up and get this stuff done together, I think that makes your team a lot more resilient. As you said, I mean, even starting out, you guys got fired, and then you were resilient, right? You came together, worked together. I think that collaborative aspect can often be overlooked, and I think that you and your team have done such an amazing job of fostering that environment, and it's a big testament to your success as well. And I think that's everything we wanted to touch on today. Dr. Tom Madden, it's such an honor to be able to be here and interview you. You're a true leader in this field, and we're all on behalf of the neuroscience program, we're all very thankful for the work that you've done and for being here today.
You're welcome. Thank you very much.
Thank you.
Thanks.